Okay. Hi, everyone. We're going to be presenting on enzyme mechanisms and reversible inhibition while also applying these topics to the lysozyme mechanism and serine proteases. So a few basic catalytic strategies are used by many enzymes. The four listed here are sort of the most common ones that we're going to see. And all of these types of catalysis ultimately achieve the same goal, which is to decrease the activation energy of the reaction and increase the rate. Oh, okay. Well, that's annoying. Um, sorry.
No worries. Um,
Frank, do you want to try sharing? I apologize. That's really
Yeah,
annoying.
sure, that's okay.
Sorry.
Um, um,
okay.
Awesome. Okay. Sorry about that. So the first type of catalysis we're going to be looking at is covalent catalysis. So many enzymes contain active sites with catalytic residues that can form temporary covalent bonds with the substrate. So that's what this diagram is showing here. The covalent bond is shown here by the red circle, where the reactive group on the active site is attracted to the substrate through a lone electron pair on the X that's shown here. This covalent bond is, this covalent bond is essentially keeps the substrate in place on the enzyme. And at the end of the reaction, the covalent bond must be broken to regenerate the enzyme. And then the next type of catalysis we're looking at is general acid-base catalysis. So it's basically catalysis in which a proton is transferred in the transition state and the reaction is catalyzed by an acid or base. So active sites may contain residues such as histidine, which is highlighted here, that can participate in an ion transfer. And by transferring a hydrogen ion, the active site may activate nucleophiles required in catalysis or stabilize charge groups or increase electric interactions that may stabilize the transition state. So in this table here, we can see that the amino acid residues that act as proton donors or acceptors, again, depending on their specific pK and what pH the reaction is taking place in. And then the next catalysis we're looking at is metal ion catalysis. So it's basically when metal ions can stabilize the increased electron density that can develop during a reaction. So that's shown in this picture here. This zinc molecule is sort of stabilizing this molecule and its electrostatic interactions. So metal ions can also provide a powerful nucleophile at a neutral pH by increasing the acidity of a nearby molecule such as water. And they can also increase binding energy by binding directly with the substrate. And then the fourth and final type of catalysis we're looking at is catalysis by approximation and orientation. So many reactions require two substrates, so the rate of these reactions could be considerably enhanced by bringing the two substrates closer together in proximity and in the proper orientation on a single binding surface of an enzyme. And active sites essentially provide a microenvironment that brings these substrates close enough together so that collisions occur. And then an example that we're looking at is a serine protease, which is, it's also known as serine endopeptidase, which is an enzyme that's responsible for the degradation of proteins, so their amino acids can be recycled for the synthesis of new proteins. This provides a really good example of a catalysis of two catalysis mechanisms, covalent catalysis and acid-base catalysis. And an example of a serine protease is chymotrypsin, which is found in our digestive system. So these enzymes cleave proteins on the carboxyl side of large hydrophobic nonpolar amino acids by a hydrolysis reaction, which is basically just the addition of a water molecule to a peptide bond. So looking at the binding site of a serine protease, it contains what we call a catalytic triad, which consists of three amino acid residues, histidine, serine, and aspartic acid. It's located on the active site of the enzyme, and they are actually located pretty far away from each other in the primary sequence if it was just one linear sequence, but due to specific folding, they're actually close in proximity to one another, and that's how they're able to interact. And each of these residues performs a specific role in catalysis. So looking more specifically at the catalytic triad, serine has a hydroxyl group that acts as a nucleophile that's actually going to attack the amine on the histidine molecule. So the histidine is going to accept a hydrogen from that serine hydroxyl group. And then aspartate is going to hydrogen bond with the nitrogen, the other nitrogen on the histidine molecule. So this is the overall serine protease mechanism. I'm not going to go over the whole thing in detail because I believe the professor will, but the important takeaways from this are that histidine and serine from the catalytic triad directly interact with the substrate and initiate catalysis. It's also important to note that there are two tetrahedral intermediates that you can see here, and those are those play a very important role in the catalysis reaction as well.
Okay. Sorry, I was trying to unmute myself. So another example of using this catalysis is lysozyme, which prevents foreign bacteria from entering our bodies who cause disease, whose cell walls have peptidoglycan, which is a half carbohydrate, half protein. And the carbohydrate part has is a repeating units of NAM and NAG connected by beta-1,4 glycosidic bond. And lysozyme job is to break it and split NAM and NAG. But they must have NAM-NAG ordering instead of having a NAG-NAM order in order for the lysozyme to work. Once being in contact with enzyme, NAM part turns into a half-chair conformation. A mechanism of lysozyme is an SN2 mechanism, meaning that aspartate has to be below NAM and glutamate has to be above NAM in order to assure a backside attack and leaving group of NAG. And glutamic acid protonates to break glycosidic bond, and aspartate's oxygen is having a favorable electrostatic interaction with the hydrogen and anomeric carbon of the NAM, assisting to form an oxonium ion. So right part becomes a leaving group, and aspartate makes a nucleophilic attack to the carbon to form the covalent intermediate and further change oxonium ion to a chair conformation of glycoside. As the NAG product leaves, water comes into the active site and protonates the glutamate, which is base now. And a lone pair of the oxygen from the water attaches to a carbon and pushes the glycosidic bond back to the aspartate, like showing here. And then the final product is that there are two final products, and NAM and NAG are separated, and the cell wall of the bacteria is disintegrated by the lysozymes. And then let's dive into the inhibitors, which is to decrease enzymatic activities. One of the two types is reversible inhibitor, which binds to enzyme non-covalently and rapidly dissociates from it. And there are three kinds, which are kinetically distinguishable: competitive, uncompetitive, and non-competitive. Competitive inhibitor binds to enzyme's active site and decreases the proportion of enzyme bond to substrate. It can be overcome by sufficiently high substrate concentration, according to this graph, this plot. And uncompetitive inhibitor binds to ES complex, the enzyme substrate complex, and makes the activity never reach the maximum rate. And just like the non-competitive, but non-competitive inhibitor binds to both ES complex and the free enzymes. And the second type of inhibitor is irreversible inhibitors, which involves slow dissociation from the enzyme because of its tight binding to the enzyme. And it is a tool to clarify enzyme mechanism by modifying functional groups. Important drugs like aspirin and penicillin are their examples. And there are 4 kinds of the irreversible. By the way, that's the kinetic scheme of the irreversible inhibitor. And there are 4 types of the irreversible inhibitors. Group-specific reagents can change the specific R group of residues. Affinity labels have a similar structure with the substrate and can modify the active site residues. Suicide inhibitors bind to enzymes as a substrate, and transition state analogs block the active site of the enzymes. Oh, by the way, these are the examples. An example for group-specific reagents, DIPF, and TPCK is for affinity labels. Penicillin is a type of suicide inhibitors, and the boronate mimic is for transition state analogs. So to sum up, there are 4 basic strategies, and serine proteases and lysozyme are examples of covalent and acid-base catalyses. The other 2 are metal ion catalysis and catalysis by approximation and orientation. There are 2 types of inhibitors, reversible and irreversible, and they differ in how tightly they bind to enzymes and dissociate from, and how fast they dissociate from the enzymes. And that's it for our presentation, and these are the sources we used in this presentation.
Awesome. Thank you, guys. Good job. So